give you a brief run through of today's events. So there's a little bit of content we have to cover. Uh, and after that, we'll be doing a coding session on Rosalind. So Rosalind is a site for bioinformatics. So before that, just in case some of you are not aware of how to register on Rosalind, I'll show you right now. Okay. So I hope you can see my screen. Can you see my screen? Okay, so that's great. So in case you have not registered yet, you can, one moment. I'm sorry, one moment. Okay, so I hope you can see this. You've at least gotten to this point. In case you have not registered yet, so you still have some time because we'll not be starting Rosalind immediately. We'll be starting it later on. So once you've come over here, you can go to the top right corner. You'll see a button for register over here. They'll ask you for your username, email ID, and a password, which you can give. After that, they will be sending a confirmation mail to your email ID, which you have put over here. So you'll have to approve it from there. Just so you know beforehand, there's a chance it goes to your spam mail. So please check that once as well. And once you're done with that, it will take a few minutes, but then Rosalind will be able to log you into the site and then you will be able to solve the problems. So in case you have not been able to do this so far, just please do this now, else it might take some time later. So has everybody understood? Okay. Okay. Okay, so that's great. So I'll leave you with that for now. For now, we'll go to the first part of the class, and that is the final part of the presentation. Just one moment. Is everybody able to join the class? Or is anybody having, I mean, UV Bari, you are in the class right now, right? I'm assuming you have been able to join it now, right? Okay, I think you can. So, okay, so let's start now. So can everybody see the presentation and the slides? Yes, okay, that's great. So this is finally episode 3 of the SARS Wars, The Revenge of Humans. So first, we'll be looking at India's two vaccines. Those are Covishield and Covaxin. And next, we'll be looking at what is bioinformatics. So you all have a proper idea of what this coding session on Rosalind means. Just before that, I want to find out. I will assume most of you are vaccinated over here. So which vaccine did you receive? Covishield or Covaxin? Covishield, okay. Shield, Covaxin, Covaxin. Okay, so actually there seems to be a fair distribution. Around half of you have taken Covishield, half of you have taken Covaxin. Okay. So Covishield people, are you all aware of how your vaccine worked? Of what exactly was present in the injection that gave you immunity? So I'm seeing this quite a few of you. So Male to Sugi, Gorinja, anybody knows what Covishield was comprised of? No idea. Okay. Okay, so no worries. If does anybody in the Covaxin group know what was in the vaccine? Okay, no. Okay, no issues. So we'll be covering that today anyway. So we'll be covering both vaccines. So you can finally find out about what they were actually doing a few years back. Okay. So one moment. Okay, yeah. So some of you have put inactive pathogen slash inactive virus. So that is correct. That is what Covaxin was made of. So we'll be getting to that in a while. Can everybody see the presentation now? Okay, so that's great. Hmm. So first, Covaxin. So, does anybody remember the AstraZeneca model that we had discussed? Viruses with vectors model that we discussed yesterday using an adenovirus. Because we had covered a few vaccine mechanisms yesterday as well. So, can anybody explain the adenovirus vector example? You can put it in the chat. Okay, so that is actually correct. So they used an adenovirus as a vector, and they took the spike protein of coronavirus, inserted it inside this vector, so it would deliver it to our DNA. And yes, so that's exactly what Covishield did. Covishield was the brand name for the vaccine of AstraZeneca, and in India it was produced by the Serum Institute of India, Pune. Okay, so if you just recap, how it does is it will take an adenovirus vector, and this vector, this virus, it causes flu in chimpanzees. So it took this particular form of the adenovirus, it genetically engineered it, so it left it harmless, but instead of that, it delivered the gene coding for the spike protein into the cell. Okay, the spike protein of the coronavirus. So this was injected into humans. So this was injected into humans so that it could stimulate the immune system by producing the spike proteins inside the body so that the necessary antibodies could be developed. So here's a brief overview of how it worked. You have the coronavirus spike protein over here. You will insert that into the vector. You will take the vaccine, inject it into human cells. The spike mRNA will be produced, which will produce the spike protein. And since the body will detect the spike protein, since the immune system does detect it, it is possible for it to fight it and produce the antibodies, though You should also remember that this is only the spike protein you're producing, not the entire virus. So it's not as though it's fatal enough to kill the human because the adenovirus has been engineered to be safe. So there's no risk of producing harmful proteins. It's just the spike protein so that the body knows that we have been attacked. It has been attacked. So just some stats actually about Covishield. So the Serum Institute of India, it is actually the largest vaccine manufacturer in the world. And during the pandemic, it was producing 5,000 vaccines a minute. Okay, so that's pretty high. That's a very high rate. And it currently produces. Oh, sorry, is anyone trying to ask something? Okay. So yes, it was the largest vaccine manufacturer in the world, and finally, it started giving its doses on 16 Jan 2021. So that was how Covishield worked. Covaxin was the second vaccine that India approved. Okay. So, and one thing, Covishield, it was developed with a model of a foreign institution, but Covaxin was made entirely in India by Indian organizations only. So there were three that were involved. The first one is Bharat Biotech. Okay. Second one is ICMR, the Indian Council of Medical Research, and NIV, that is the National Institute of Virology. So if you remember that we had discussed yesterday on Wednesday, that the vaccines which we're looking at, a lot of these new vaccines, they don't use the conventional style, right, of putting a dead virus or weakened virus. They don't usually do that. We looked at, for example, DNA, RNA, and similar genetic vaccines. That's the point. We weren't looking at old vaccines, genetic vaccines. But Covaxin is actually it's an old type vaccine. So if you look at it, it will actually inject an inactivated version of the virus. Okay, it's a killed version. It cannot harm you, but it will still cause your body to produce antibodies. It is a safe vaccine. There's no risk of contracting COVID because the virus is inactivated. It's dead. I mean, but it is good enough um, as a vaccine. So it was approved in November 2021 for emergency usage, and it was the first vaccine to gain this status, Indian vaccine to gain this status. It was approved by the WHO, the World Health Organization of the UN. And by January 2022, 13 countries would approve it for usage. Okay. So is everyone following so far? Have you all now understood what was actually injected into you during the pandemic? Yes. Okay. Okay. So I hope that's clear. So we'll move on to our next part of the session, that's bioinformatics. So first I want to know how many of you understand bioinformatics or you're aware of what bioinformatics means? Oh, okay, wait, I will share the link one moment. And just a reminder, you will need your laptop for this session because you will be doing coding on it. So just so you're aware, if you're having any issues, I've posted the link in the chat for some reason, so you can try that. It's also in the WhatsApp group. Okay. But in any case, is anyone aware of what bioinformatics means? Yes. So sequencing genetic information through coding. Yes, that is correct. So, um, what you're doing in bioinformatics is you'll have noticed that I've used the word code a lot of times during the session, right? So you're talking about a DNA code. You're talking about an RNA code. You have codons which are coding for the protein. So you're giving some sequence of nucleotides, right? A, G, C, T. And if you're talking about RNA, then A, G, C, U, right? So the point I'm making is you have these genetic codes, these biological codes, but it is possible to use computer programming to develop this, to understand this further. So you can use coding. So what happens is often you have very large data sets. By data sets, I mean maybe you've been given a long strand of DNA. You've been given the sequence, but it's too large for you to just read it and make something. It can be thousands, 10,000s of base pairs long. I mean, if you're talking about DNA in humans, that is around 3 billion base pairs. And it's not possible for a human to be able to just look at that and understand it, right? So you will have to use computer science tools to understand that. And thankfully, it is very convenient because that's how our biology is. It is possible for us to convert it to a computer. So here's what bioinformatics means. So we have a lot of codes, okay, in biology or biotechnology. So to be able to study this kind of information, we go to the field called bioinformatics. So it helps us make some sense of biological data. So for example, we're aware of DNA, it's transcripted to RNA. Okay. So say you're given a strand of DNA and you're asked to convert it to RNA. You can't just sit and just try to convert it alphabet by alphabet, right? Because as I said, sometimes these sequences are very long. It's not possible to just do it manually. So we use computer coding, we use computer programming, which can transcript it, and that helps us. It makes our task a lot easier and actually helps us understand something that we're given. Else you're just looking at something and you have no idea what to do with it. So did everyone understand it now, what bioinformatics means? That is yes, right? Okay. Okay, so everybody seems to be clear. Okay, if you have any doubts, just again, you can put it in the chat. Now we'll be moving on to Rosalind. So I just want to be 100% sure. Has everybody logged in and has logged into their account on Rosalind? If you have done it, please type 1 in the chat. If you have not been able to do so, you're having some problems, please put 2 in the chat. So just please put 1 or 2. Okay, 1. Okay. Oh, okay. So I have the link in the chat, but okay, I'll just put it once again. Give me one. Okay, I'm sorry. Yeah. So as I was saying, you will also have to be writing the code. So if you're not aware of any compilers, you can use the one that I've just put in over here. It's called Google Colab, in case you're not aware. So you can use that. And the first one, that Rosalind, that is where you can access the site. So I see that a couple of you are having issues. So have you sorted it out now? Shubhankar and UV Bari. Okay, so where are you having an issue exactly? Or is it just waiting, or are you just waiting for it to sign you in? Okay, so if you um, just tell me if you're having an issue. So we'll move on now. But just in case you're worried, this session will be recorded. You can refer to it later so you can understand what we're doing. You don't have to be worried about being left behind or anything. And just one more thing, it's possible that some of you are not used to programming. This may be your first time. So you don't have to worry about that. We're here, like me and the other coordinator, so we can help you a little. We can help you and guide you over here. In case you're not able to, we will be sharing some resource material that will help you understand what we're doing over here. So don't worry. And you always have the session recording as well. So you can refer to that as well. And one last thing, some of you had asked me doubts on Wednesday, and I had said that I would get back on it, get back to you later. So I will be staying back after class. So I will on the same meeting. So you can stay back as well and ask me what you had been asking. So I will be able to answer that. In case you want to listen to it, you're of course free to stay back. And if you have any new doubts to ask, please also stay back because I will be going through that. Okay. So I'll just wait 1 minute and then we'll start. Okay, so now let's start. So this is what the site will look like. So you'll just in case you're at the home page. So I'm sorry, wait. In case you're at the home page, you will see a problems written on the top left side. So click on that, and below problems, click on list view. So you will see list view over here. So click on that. Is everybody following? And is everybody at this page? Put a one or two in the chat. Just one if it's good for you if you're at that point, or two if you're lagging behind. Yeah. Okay. Okay, so everybody seems to be following on, which is great. So here you will see a list of problems. So they have titles also DNA, RNA, etc. So let's go to the first one, counting DNA nucleotides. Hmm. So what they've done over here is they've explained the problem at the beginning, and then they've given you a sample problem so that you get an idea of what's happening on. So if you see what's given over here, they'll give you a string, which is a collection of symbols, okay? Uh, and they'll have some alphabets, so A, C, G, and T. So these obviously stand for the nitrogenous bases. So what they're doing is they're giving you a DNA string over here, and they're giving you a DNA string, and what they're asking you to do is asking you to give four integers. So the first one will tell you how many A's are present in the sequence, the second for the C, the third for the G, and the fourth for the T. Okay. So for example, we've been given this sequence over here in the sample data set. So AGC, this is given, and they've given 40. So 20 means there are 20 A's over here, 12 means 12 C's over here, 17 G's over here, and 21 means there are 21 thymines or T's over here in the string. So is everybody following what the problem means or what it's asking you to do? Okay, so you've gotten the problem, but now you have to write a code, right? So if anybody's aware of how to write the code, just you can do it yourself, but please follow me if you're not aware of how to do this. So has everybody opened Colab now? Google Colab. Oh yes, I'm aware my screen is not sharing. Yes. I've stopped it for one moment. I'll be coming back. So has everybody opened Google Colab? Okay. Okay. So I hope everybody's on Colab now. So what to do is I'll now share my screen. Okay. So you will be on this page, and on the left hand side you will see a plus code over here. So just click on that. Hmm, hmm. Um, has everybody got into this point now where you can start writing a code? Yep. Oh, you're not able to see my screen. So is it one second? Wait, I'll stop sharing for one moment and I'll resume it. Okay, so now can you see it? Oh, still no. Um, just confirm once last. If you can see my screen. I'm on Google Colab. Now because I'm screen sharing, so just tell me if you can see it finally. I'd stopped for one moment in the middle. Oh, it's just blank. Okay, just you can try leaving and rejoining once if it works. If it does not work, there's the session recording which you can look at. Okay, so just try leaving it once. Okay, and just check your network issues. But okay, even if you are not able to see, it's fine because there'll be a recording, and in case you're not understanding something, you can just message me. Okay. So I'll start writing the code over here. I'll first write it, then I'll explain how it works. Okay, so for those of you who are aware coding, you'll probably be aware that I want to write count underscore c is equal to count underscore c plus one. Is there a fancy way of writing it which you're aware of? Which you can tell me now. Okay, that's great. You can see my screen now. Nice. So is anybody aware of any other way of writing that? Count c is equal to count c plus one. Okay, it's fine. That's not an issue. We'll get to that. Okay, so can everybody see the code that I've written over here? Okay, okay. Sorry, I didn't see. But yeah, you're correct. So you can say count underscore c plus is equal to plus equal to one, or you can use plus equal to, yeah, that's the same thing. Eco and 2 3 So yeah, that's right. That means the same thing as I've written over here. So I'll show you how that would work. You can look at Okay, so this county I've written it in the kind of cool format of computer programming. So you can also write it like that. Of course, you can also write it like this, but this is just slightly shorter and more convenient. Okay. So is everybody able to follow on to the code? Can you see the code? I mean? Okay. I've not explained it so far. I'll be explaining it now. I just want to make sure that everybody can see it so far. Don't worry, I'm not. I'm explaining it only. You can't, oh wait, I've blocked it for now. Wait, you will be seeing my screen now. Is it visible now? Just put it in the chat. Okay, you can see it now. Okay. So, okay, that's great. I'll now explain this how I wrote the code exactly. So just a reminder of the problem. It's giving us a DNA sequence and we have to count number of times A comes, C comes, G comes, and T comes. So, okay, just let's understand how we're doing it in our mind first. So we have the string, okay? We look at the first alphabet, that's A, okay? So initially there were no A's which we had noticed, but now we're noticing one A. So we'll just think, okay, I'm keeping a counter for A that's gone from 0 to 1 now. Next alphabet you see is G. Now you'll keep a separate counter which is for G. So you'll say, okay, G initially was at 0. Now I have one G that I can see over here. Same for C. C you go from 0 to 1. And same for T. You had 0 T's initially. Now you've seen it, you'll increase the counter for T from 0 to 1. So basically you're keeping four separate counters, one each for A, C, G, and T. And whenever you see it, you're increasing the counter. And by the time you're done, you'll have seen there's 20 A's. So you'll write it like that, 12 C's, 17 G's, and 21 T's. Okay. So that's what will happen in your how you'll do it mentally, right? So you have to do the same thing in a computer in computer coding, except you'll have to simplify the process. Like you'll have to simplify the logic, and you'll have to write it like that. So first you'll see I've written something called code is equal to. So this is basically storing the string. Okay, so the string is the variable, which is the DNA over here. It's a long sequence of the alphabets. Okay, so that's what code means. I've kept four different counters over here: count underscore A, count underscore C, underscore G, underscore T. These are the four counters for each of the bases. And there's um, yeah, so you'll have four different counters. There's zero at the start. Okay. Now what I'm doing is I'm running a for loop. So what this for loop will do, it will run through each alphabet in the code one by one, and it will put it through this sequence that I've given over here. So first it's saying if the base stands for each alphabet in the code. So if it's an A, you'll increase the count of A by one. If it's a G, else if it's a C, if it's not an A, but if that alphabet is a C, you'll increase the count of C by one. If it's G, G by one, T and T by one. So by this, you'll be able to count all four of the nucleotides and how they're coming in. And as you can see over here, you have to print it, right? So you'll just put a print function and you'll type the four integers and it will print it. So I'll put an example over here. So I'll take the sample data set which they had given since you also know the answer for it. So you can copy that and put it over here. Okay. And once you're done with it, you can just, there's a small triangle over here that's for running the program. Oh, okay. Okay. Yeah. So it's run the program now 20, 12, 17, 21. That's the same answer that they're given. So we can see how the program works over here. So has everybody got the point now? Just put it in the chat. Please write the code once so that you get an idea of what's happening. Okay, and just run it again using the sample data set, which you'll see in the other end. I'll just give you 1 minute. Yeah. Oh yeah, yeah. You have to go to the problems. I'll just share my screen for 1 minute. Yeah. So you're here. You'll see a list of problems over here. So just click on counting DNA nucleotides for now. You'll see um, problems. You'll see a list view. Just click on that and you'll get to this page. And counting DNA nucleotides. Just let me know once you're here. Okay, mail to Sugi. I'll just look at your code 1 minute and I'll let you know what's happening. Okay, if you just look, you're supposed to, you've written, when you're initializing, you've written COUND underscore G is equal to 0. You've written count instead of count. So, and later in the for loop, you've written count. So that's why it's not recognizing the variable. So just change the count to count so that the problem will be sorted then. Yeah. Okay, so you have to write the code. The screenshot is in the chat. It's on a site called Collab. I'll put the link. I'll put it again. Just give me 1 moment. Okay, so Khushi, I've put in the chat now. Just check if you can see it again. Or you can write the code over there. Just if is anyone having any errors in their code now? Just let me know now, please. Okay, so I hope it's working for everyone now, and everybody has been able to do it. Just in case, no, if you are having any issues, just again, I'm reminding there will be a recording of the session. You can view that again. Okay. Now, just moving on. One question though regarding this problem: how can we use it? How what's the practical application of such a problem where you are counting the number of nucleotides in the sequence? So, how can somebody give an example of that where this can come in use in real life? Aditi, just one thing. When you've written code at the start, if you write code is equal to, you've just written code and you start the code. So just change it to that. Uh, Aditi, so just what you should do is you've written code, double apostrophe, and then you've written the code AGCT or whatever. What you should write is code is equal to double apostrophe, and then you write the code. So did you get it now? Okay, so that's good. Just one more. Okay, so 2 3 F, yeah, that's what you have to do. You're in Python Village, I think. So wait, I'll share my screen so you know where to go. Yeah, so I think you are in Python Village. Go to Bioinformatics Stronghold over here. In problems only, Bioinformatics Stronghold. Then you will see it. Have you sorted the issue now? Okay, so I think you've gotten it now, so that's great. Okay, so few of you have put the answer. For extra or less base pairs, genetic disorders, abnormalities, yes, that's correct. So you can you can look at the sequence and you can although we're counting the number of nucleotides over here, you're not exactly looking at what sequence they're present in. So how would you check that? I mean, that's a different problem you're talking about actually. So how does the number help you over here exactly? Aditi, after you've written for base in code, put a colon. So write it like this. I've put it in the chat. You just put that colon mark. And it's the same after if. So after you've written if base is equal to a, put another colon mark over there. The indentation also. Yeah. Indentation will come once you've put in the colon marks. That's not an issue, but yeah. Okay, so wait, I'll just look at the answers in a minute. So Khushi, the elif that you have written, if elif base is equal to C, you've written that under if. So what I wanted to do is you just go to the elif and go before the E and press a backspace. Okay, because it should be at the same. If you see elif, it's written ahead of if, right? So elif should be at the same line as if. If you see at my code, if you look at the code, did you get it now, or should I present my screen once? Yeah, same indentation, yes, sir. That's how you. That's there's a space you've given after elif, which should not be there. Um, I'll just put the link again. Just tell me if you're able to use it now. Just give me one second. Okay, so Rakesh Shailaja, outside the print you have two brackets right over that sign. So after the second bracket you've put a double apostrophe. Try removing that once and check the code again. And one more, and I'll put the link for collab once again. Just give me one second. Oh yeah, try that. Try this link now. Okay, so NG, I think that's good. Yeah, you've gotten the code right. So that's great. Anybody else is having any issues? Okay, so I think everybody's got. Wait, one sec. Oh, sorry. Wait. Oh yeah, so UV barriers have to go to Python village. Miltusuk, you've gotten the correct answer. I think you just wrote. I think do you write a slightly different code so that it would give you step by step? Oh, okay, wait. I think okay, I understood what you've done. So the print you have put it inside the for loop, I think. So what you should be doing is you should put the print and the for at the same indentation. That's why it's coming as a repeated printing. So you can do this also. It will give you the correct answer. It will give you the same answer at the end. But it's a very long answer, so you can just put it later. Oh yeah, sorry. I think I said the wrong thing. So go to bioinformatics stronghold. I'm sorry. Made to yeah, okay. Just look at the print. It should be at the same level as for, not ahead of for. Otherwise the answer is still correct. And Aditi, you've written print with a capital P, so put it with a small p. Sanjana, once you written elif basis equal to C, just put a colon. And same for G and T. Put a colon over there. Oh yeah, Sampurna, if and elif should be ahead of for, and they both should be at the same level. Okay, that's great. You got the answer. Nice. Also, you can of course ask me just one more thing. They sometimes give you a hint also at the bottom if there's any error. So you can try to look at that and see if you can solve it. If you can't figure out what it's saying, you can just of course put it in the chat. And feel free to put it in the chat in any case, if, regardless of anything. If you're having any issues, just put it in the chat. Okay, so I hope everybody's done with this. If there's any issues, you can just refer to the recording once again, or you can refer to the screenshot as well. I'll just share my screen again so you can just check once if you can now finally see it. Oh, wait. Okay, so you will be over here. Go in problems, list view. You will see it over here. If you are in Python Village, don't be there. Go to Bioinformatics Stronghold. Okay. Anyways, if you are unable to find, just look at it again. Anyways, we'll move on now. So it asks you what's one important function that this kind of a code does. So I think somebody had given the right answer. So Sampurna had said to know thermal stability of the DNA, and if GC content is more, it's more thermally stable. Yeah, so that is one thing. You can figure out GC content from such a code. And okay, so that is correct. And one more thing is for identification purposes. So let's say you put the some DNA sequence in the code, and let's say figure out that it has 50% A. It has 50% A, and remaining 50% is C, G, and T combined. And let's say checking for the presence of a virus. Let's say coronavirus. Now let's say you know for a fact that coronavirus does not have 50% A content. So you don't have to run any further tests. You can directly rule out that this is not coronavirus, right? So that's one way you can use it. You can use it as a preliminary test to just check if this is the organism you're looking for. So that's one kind of basic test you can use it for, just a pre-test in a sense. And just one more thing that I want to ask you. So what happens when you're trying to physically, like biologically in a lab, trying to analyze DNAs? You will do something called gel electrophoresis. So you'll put the DNA in a gel basically, and it will be separated. It will be cut, and on the basis of the size, it will be arranged in a random pattern. So there's a there's a possibility that a small part of the sequence is repeated accidentally. Or you may, for example, I'll give you a sequence and I'll explain from there. Okay, so you have these two codes, so they're different, obviously, as you can see. But you'll see that the GCT, it's repeated two times in the first one, while it's only repeated once in the lower one. So here's the thing. When you're analyzing the DNA, you can only look at what that small strip of DNA is doing. You can only look at that small piece. It's kind of the smallest you can look at. So how can you know whether or not a particular sequence, it can be repeated, it can be present in two parts, the previous one and it can be present in two pieces at the same time. So how can you figure out whether or not it is just one sequence that is repeated twice, that GCT, whether it's been repeated twice or whether it is only present once? Okay. Okay, so you can again use it for motif analysis also. So what that means is you're trying to test for the you're trying to test for the presence of a particular protein or a gene for that protein over there. So yeah, you can use it for motif analysis also to just check whether you're on the right path, whether it matches the data that you know for the particular sequence that you're looking for. So you can use it for that purpose as well. So yeah, that's right. Okay, so we'll just do one more problem now. Oh, and just one thing, I'll tell you how to answer the problem over here. So you've gone into counting DNA nucleotides. So if you have solved the if you've written the code, what you can, if you've logged in, I'm sorry, you'll see a option at the bottom of the page called download dataset. So click on this download dataset. It will give you a long code. Okay. So they've given this for me. You just have to copy that. Control copy. Put it in your code. Oh wait, I'm sorry. 1 second. Okay. So from Rosalind, you'll copy the data set. You'll have to click over here. It will give you a data set at the top. Yeah, so you'll see it over here. You have to download that data set. You have to copy it. You have to take it to your code which you have created. Insert it over here, and you have to solve it. Just be careful because you have to put the code within double apostrophe, right? So there's a chance that the code by some mistake, it is not enclosed by double apostrophe comma. So just check that once, especially if it's a long code, it sometimes is not visible. So just put the code and run it again. You'll get some answers. So just copy that. So for me, it's these four numbers: 208, 226, 226, 222. Control copy. Go back to Rosalind. There'll be some space at the bottom. You can copy your answer here. Control V and submit. Okay, and it will tell you whether you've solved the problem correctly or not. So you can do that. So anyways, we'll move on to the next problem. That's transcribing DNA into RNA. So what they've given over here is they've given you a DNA string. You have to convert it to RNA. Okay, so that's pretty straightforward. So just one question though. They've given you the coding strand over here. So can anyone tell me what's the difference between how do you convert the coding strand to the the coding DNA strand to the RNA strand? Don't have to write a code, just tell me in the chat, like in simple words. What do you do to it? Okay, so just one thing, it's not reverse. I'll tell you why. It's the coding strand is the same sequence almost, just you're replacing T with U. Okay. Navya, it's not the reverse strand. If it was the template strand, it would have been the reverse. Then it would have been reverse. Here it's almost the same, just you're replacing thymine with uracil. That's the only difference. Okay, no issues, no issues. That's fine. Yeah. Uh, so this is the template. Sorry, coding standard. This is the coding standard talking about. Yeah. Oh yeah. We're doing the second problem. Yes. So once again, if you're writing another code, wait, I'll show my screen. So for writing another code, you can keep the previous code. You can press on plus code again, and it will give you some space to write second code. Okay. So I think you've gotten an idea of what the first code was. So I'll not share my screen with you. You can just write the code yourself. If there are any issues, of course, you can write it again. I'll just tell you a few things that you should know related to the syntax. Syntax is basically the rules of the language which you're using over here. So we're using Python over here, just in case anybody is wondering. So this code it basically means you're defining a string, which is a long sequence of alphabets or characters put together. So here it will be in TCG, some format like that. And on both sides of the sequence, you'll have double apostrophes. Okay, remember to use it like that. This count underscore review, you don't use it needed for the second problem, but that's just an integer which initially its value is 0. We can change it later. So when we're saying for base import, so code is this sequence. Base means it's some kind of a variable. Variable as in what I'm saying. It will first analyze the first character of this code and it will put it through this sequence, this code, I mean to say. Once it's done with that, it will move on to the next alphabet, then the next alphabet, and so on, once until the entire code is done. Okay. Inside, you can write it in the same format. Just one thing: when you're writing if base is equal to a, put two equation signs. The reason is you're comparing two values, right? You're comparing base and you're comparing a, capital A, right? You're not saying base is equal to, you're comparing them. So put a double equal to sign. But when you're adding it, count underscore is equal to, just put a single equal to sign. Okay, don't put double over there, put single over here. So just keep this in mind. And a small hint for problem two, you have one code for the DNA sequence you're given, put one code for the RNA sequence. You can say code underscore two or use something. So this is a small hint, or you can try it yourself and just let me know if you're having any issues. Wait, how you pasted that long code? 1 minute, I'll share it. So go over here. You have to click on this download dataset. It will come over in your download. So you can see it over here, Rosalind 4. Click on that. They will have given that long sequence. So just select the entire code, copy it and paste it. Control C, Control V. Yeah. Did you get it now? Also, there's a time limit for the dataset, so just do it quickly. Okay, so I'll just write the code now. I'll be sharing my screen if anybody wants to look at it. Of course, just you can try it yourself also. Just in case you have not entirely gotten the hang of it, I'll show you once. Okay. Oh, wait, you don't have to paste the code. You don't have to paste the code. You can just take the answer you're getting and you can paste it over there. You can paste it. They'll have given you some space over there. You just paste it over there. You can do the code also if you want, but the answer is also enough. Okay, I'll just show you my code again, just so you can get a look at it. Hmm. This is the code. There'll also be a screenshot in the chat. You can look at that also. Okay. Fahad, did you also just verify that the code is working well? Navya, you could also do that. Yeah, that is simpler, I agree. Yes. Okay, Fahad, that's great. If you're done with the code, you can also just, of course, go on Rosalind and download the dataset and do the problem. Okay, so the third problem that is slightly tricky. I agree. I got what's happening. Just I'll let you know how to do that as well. Just give me 1 minute. Let a few more people get there. Okay, so I think you've gotten a basic hang of Python or programming now. So we'll be uploading assignment 2 later on. And for that, we'll be giving you the required syntax and we'll let you know more about the assignment of how you're supposed to solve it. So please do that. You'll submit it alongside assignment 1 only. The deadline, we will let you know about that also. So if the session is over for now, if you have any doubts, you can ask me. And I think some of you had a few doubts yesterday, which I could not get to. So please, just feel free to stay back and ask them now. If anybody else is interested in hearing this or wants to ask other doubts, you can stay on again. Just give me one moment. I'll share the third one also. Okay, thank you. Okay, and just please also let me know how you found these three classes of SARS Wars. So like it was pretty great. So just thank you everybody. You were a pretty interactive audience. So it was nice to have everybody answering whatever I was asking, and it was great to see people asking a lot of doubts, pretty high level doubts as well. So that was great. So thank you. I hope you enjoyed it. The session is over, but I will be taking a few doubts now. So if you want, you can stay back for that. And yeah, I'll be sharing the third one also. Okay, so thank you, everybody. Just a reminder, some people had asked me doubts yesterday about taurine, apetamers, selects. So, a lot of topics. So, just if you have any doubts regarding that, just please stay over here because because I have to answer. I have to give the answer for that. So, yeah. Okay, so thanks a lot. It was great timing. So happy that you learned coding also. Oh, hope you liked it also. So yeah, just give me 1 second. I'll look at the doubts you're asking. Okay, wait. You're still not able to paste it. So just to understand, are you not able to paste the data set or are you not able to paste the answer you're getting? Oh, okay. I'll just show you once again. Just see if you're able to follow on now. Okay, so you've gone to, you can see my screen, right? Just let me know that. Yeah, you can see. Okay, good. So you've gone over here, click on download dataset. Wait. And you'll see, go and download at the top right, you'll see this downloads button over here, arrow, download facing arrow. So click on that. It will show you the latest download you've made. So you'll see a file, Rosalind underscore DNA. Click on that file. It will give you that long sequence like CTTG for your, it could be different for you, but you'll get that. So copy paste the entire thing, select it entirely, control copy. Then go to your collab part, collab, collab page, where you've written the code. So yeah, it's here. And if uh, 1 second, you have to paste it over here. So backspace, control V. Just one thing, the apostrophe, double apostrophe usually comes on the second line. So just remove it and put it on the first line itself, directly after the code. Hmm. Okay, so this is what you do. You'll run the program. you'll get some answer over here. So for me, it's 220, 220, 270, 201. So I'll copy this answer, Control C. I'll go back to Rosalind. They've given you some space over here. So answer submission, copy your answer here. Put Control V over here. Your answers will come over here and just press submit. Um, so did you get it now? Oh, you're unable to download the unable to open the download data set. Did you get where to do it from? Or where to click the button? You can't paste in collab. Okay. Wait. Wait, I'll solve one problem at a time. You can't paste it in. Okay, so after download data set, just go in your downloads page, you'll open the document Rosalind underscore DNA. You can open it. You can copy the code and paste it in your, you can, sorry, copy the sequence of DNA and paste it in your code and do the problem. But you're saying you're not, like, saying you're not able to paste it in Colab. So, Sanjana, the reason is because it's not wrong, but what's happening is, okay, actually don't do that because it will give you a very long answer. Print has come under LL. So print should be at the same indentation of code. So if you see code count underscore a, they're at the same level, right? They're at the same indentation. Print is a little bit too ahead. So get it back. It will solve the problem. Just press backspace. I'll give you the code for the third problem. So just give me one moment. It will, Sanjana, that will still give you the right answer, but it will be very inconvenient. So don't do that. Just take print behind. Same line, just backspace it so it gets to, so it's at the leftmost part of the line. No problem. Hmm. I'm not sure why it's not working for you. Do you mind if you share your screen and show it to me once? Just tell me if you have a problem with that. UV, wait, buddy. Just okay. You can log out this session. Okay, I'm just telling you, convert the data, copy the data set, and paste it into the code. Um, wait, sorry. T version, Rani. Do you mind if you present your screen? Oh no, wait, I'll have to give you permission, but okay, wait, I'll just make you a presenter now. Give me a second. Check if you can present now. You should be able to do it now. Or you, this is not the assignment, by the way. This is assignment two of Rosalind. We'll be giving you the assignment later on. So you will have to fill in the code. So we'll tell you more about that later on. Or it will be coming today. So yes. We'll let you know more about the deadline. Okay, wait. You'll see a screen option next to your mic. Try using that. Try checking over that. Okay, well, if you're unable to do it now, it's fine actually. So um, you can leave it for now. Okay, so you can leave it for now. If you have any doubts, you can ask me again about day two because quite a few of your doubts then. I will give you the third problem also. Just give me 2 minutes. I'll give you that. If you have any issues, just again watch the recording and we will be sharing some resource material with you. You can look at that to understand how to write the code. The assignment also we will be sharing with you shortly. So yes, does anybody have any doubts? Just wanted to make sure from day two, day one, day three, anytime, any day. Okay, it's no problem. Okay, it was great to have everybody. We'll let you know when you have to submit it. I think somebody was asking me about taurine yesterday. So, who was that again? Wait, sorry. Do you not have the recording for the first session? It's in the chat. It's in the chat, and we've also shared some links with you. I checked WhatsApp and Teams once. Oh, yes, I will share more resources on Sherlock and CRISPR. Yes, I will do that. I'll make sure to do that. Yes. The reason you can't see an output is because you've not put code, right? If you put some code inside, then it can get an output. Otherwise, it's invisible. If you have no input, then you can't have an output. It's like that. Just put code is equal to, let's say, AGCT or any sequence you want, then you will get some answer. Wait. Echo is it echo.akaki, is it showing an error for you, or is it just giving a blank output? Can you share screenshots of your code over here? Gorija and echo.akaki. Okay, if it's lagging, then okay, give it some time maybe. Okay, that might also happen. Yes, that is also an issue. Oh, you can retry it once again and then you can reach out to me. I'll just give my number over here. You can find it on WhatsApp also, but just in case. Okay. Yeah. Yeah, output will communicate code and programming. Yeah, so that's fine. But yeah, you can reach out to me later again. So I've put my number in the chat if you have any issues again, and you can look at the recording again. You can look at the resources again. So that's not an issue. Yeah. Oh, but using a different language. Okay, so I hope most of you are done. I'll just wait for a few more minutes. After that, I'll end the meeting. Okay, so good question. The Cas proteins, they're not made from the viral DNA. It's a part of the bacterial DNA only. But this part of the DNA, it's close to the viral DNA that's stored over here. So basically, you have viral DNA. Okay, that's from the virus. You'll be storing that between multiple some sequences, and close to this viral DNA, there will be close to this viral DNA, you will find the genes coding for the Cas protein. So it's not virus. It's not the virus's DNA. It's close to it though in the bacteria. It's the bacteria only. Yes, you're right. So there's a lot of Cas proteins actually. So you have Cas1, Cas6, Cas9, Cas12, Cas13. Each of these have a lot of subdivisions. So I'll have to get back to you on how many there are exactly. Oh, okay. So in the first part, how have we put a major halt to the pandemic? I think this is from the sneak peek you're talking about. So that I was referring to the vaccines. So it covered genetic vaccines in the second lecture, like DNA vaccines, RNA vaccines, and using adenovirus as vectors. So that is how we had put a major halt to the pandemic. I was talking about how vaccines had actually helped prevent or lessen the impact of COVID, I think, on a lot of people. So that's what I was speaking about. And thank you. So it was great. So happy that you found something nice. So actually, wait, I've gotten the code. I'll just send it now. Yes, thank you very much. I'm happy to see that you found it interesting and nice. So thanks a lot. Thanks a lot for being interactive and making this class fun for me also. So thanks a lot. Yeah, I've posted the screenshot of the third code now. Yeah. Is this still the first problem you're on? We'll let you know more about the submission. I mean, the submission deadline. We'll let you know about that. Okay, so I assume you're talking about the Biotech Club projects that we're doing this year. Are you asking about that? Okay, so the sixth project, so like the one I'm a part of is just one second. Okay, thanks a lot, Sagnik. It was great to have you as well, and it was great to, I mean, have a very interactive and fun audience. So thanks a lot, and nice to see that you enjoyed it. So that's great. And yeah, the projects that I'm doing. So the first one is neurospike. So what we're attempting to do over there is you have how neurons or networks in our brains work are when a certain impulse is passed to them, when a signal is given to them, basically they will spike the the um, voltage in our neurons. They will spike up at that point, but after that impulse is gone, it will immediately drop down to its usual value. Okay, so the voltage will go up and it will come down immediately. It's somewhat like a spike or a line, if you can think about it. But if you're looking at real life electronics networks, hardware, if you look at those chips, what is happening is the voltage is always high, so the potential difference is always high, and that leads to a lot of energy is lost. It requires a lot of energy to work. And this is regardless of whether or not the signal is actively passing through it or not. The voltage is always high. So suppose we're able to take this kind of a neural networks, brain's neural network, and put that into electronic chips, it would be able to reduce power consumption, energy consumption by 1 over 800. So it would just cost 1 over 800 of the current energy consumed. So that's what Project Neurospike is trying to do. We've renamed it now to Impulse Control. So yeah, that's the aim of this project. Okay, nice to see you found it interesting. Okay, so there's more projects. So you also have a project on autism modeling. So that's what we're attempting. That's not my project, but what we're attempting to do is we're attempting to make a circuit and then make modifications to it so we can somewhat mimic an autistic mind, how it functions, how it receives inputs from the outside world. So we're trying to get that in a circuit, and then we're trying to make more modifications, more changes to the circuit, so we can understand more about autism and more about how maybe whether how it can change, how what kind of therapy can be done for the brain. So we can try to do that. So that's Project Autism Modeling. Then the next project we have is nanoparticle gun. So what we're doing over here is we're going to be creating a gun, just a nanoparticle gun. So it will be able to shoot very small particles. Purpose is to get that into cells so you can observe material. So for example, you can take fluorescent materials. You can, like we had discussed on day two, and we can inject that into the cell. Okay, using a nanoparticle gun, it can go inside. It can reveal like it can basically light up the cell, and we can observe parts of a living cell under greater detail. So it can give us a better understanding of what's happening. And a nanoparticle gun is good because it's not easy for a particle to just cross the cell, right? It needs to be going at a higher velocity to give it a good chance, which is why we are using a nanoparticle gun because it will give it a high velocity, it will give it a high speed. And using the way the gun is able to give it is using pressure difference. So we will be using helium gas and use we'll be creating a pressure difference. So using that pressure difference, the nanoparticles they will have a high enough velocity that they will be able to cross the cell membrane. So they will be able to do that. Okay. Okay, that's nice. Are you a student in IIT? Just out of curiosity. Okay, no, it's great you're noting it down. Okay, so you're not a math student. Okay, okay. Okay, no, still. Okay, it's nice that you are actually, it's great to see that you're interested in our projects. But yeah, so we have a few more projects. So one is drug microbiome. So each person, like individual, they have some bacteria in their gut, okay, in their stomach, in their intestines, and that's important for digestion, for what do you say, fighting of other microbes, like harmful microbes that might come on with your food. So you need some bacteria in your gut, okay, so that's important. And this varies from person to person. It depends also on which region you're from. So for example, we're all Asians, we're from India, but so we might still have somewhat similar microbiome is basically the network of this bacteria that we have, okay. So it might be somewhat similar for Indian people, but then you go to, let's say, Europe, they will have a different microbiome. You go to Africa, they'll have a different microbiome. Okay. And okay, so that's one thing. You have properties. It can depend on your lifestyle, your age. Okay, those are also ways that this can vary. So what we're trying to do is, and often there are some ailments that you have digestion related issues. So we're trying to get a proper idea of how first of all, you have to categorize everybody. You have to categorize the kind of microbiome, the kind of bacteria you're going to find in each person's stomach, and based on these categories, you have to figure out what kind of treatment you can give, what kind of drugs you can give. So that is one more project that we're doing. Then the fifth project, that is Open Bio. So we're constructing a library. So what I mean by library is not the books one. So it basically contains a lot of codes, and these codes, they have some specific function for bioinformatics. So yeah, so we did bioinformatics today, right? So you can see some of the codes that we wrote. So one was on DNA to RNA transcription. One was counting DNA nucleotides. So these are all uses of that, right? Um, Maybe you might want to use it in a code, but it might be a little bit inconvenient for you to just take that, right? So what you can do is you can go to a library, you can import the code and put it in your own code, and then you can just keep on writing it as on. So it's much more convenient. The task has been made easy for you. So this is what OpenBio is doing.
Is it something like the HGP project?
Some I somewhat a human genome project, right? So yeah, they
Yes.
were sequencing. Yeah, that it is somewhat related. Yeah, I would say yeah.
And is there anything to do with the geobotany?
With geobotany, I am not a part of these projects except for Neurospike. So I am not actually aware of that. I'm sorry.
No, so basically, geobotany is like where you use plants to search for minerals, which is called geobotanical prospecting. So it's like a crossover between geology and botany.
Okay, okay. So you're asking this with respect to drug microbiome, I'm guessing.
No, I was just wondering if you guys are developing any technologies to help with this or anything like that.
Um, so far, no, but actually it sounds interesting. But as far as I know, we're not doing it right now, actually.
And what about biomineralization?
Okay. Sorry, I didn't hear what you said.
Biomineralization.
Okay, okay. Yeah, so I think this kind of comes under, somewhat comes under the last project. So, like, we have Vaishnavi, she's a coordinator. So, she's actually a part of that project. So, I think it's best if you ask her that question.
Okay.
Hello, am I audible?
Yes, ma'am.
Okay, so the project which I'm working on is EnviroFuel, wherein there are some organisms which can actually increase their lipid content, or there are some microbes which have a lipid content in their dry mass of about 77 or 70 to 80% of their dry mass is the lipid content. So what we are planning to do is we are planning to use such organisms to make biodiesel.
Oh, and there was another similar project, right? Where you are using waste paper to make biofuels.
Yeah, we are trying to work on like similar lines with like yeah, we are trying to work on similar lines with that project. Yes.
And what do you people think about fungi bricks?
Fungi bricks. Okay, can you tell about it?
So it's basically people they're using the mycelium of fungi to make construction materials.
It actually sounds like a great idea. Like we'll think on it. We'll surely think about it.
Okay, last question. How do you people come up with all these ideas?
You just like keep reading stuff. So, and then later, when you remember that you have summer school, you just come up with these ideas that this is something I read, I found that pretty interesting. So I think other people would also like to hear that. So that's just how we do it, matlab. You have to find something you are interested in, think of whether that will be interesting for other people also, and then you just choose that. So yeah.
Okay, thank you for your time. I like today's session. I wish I could code properly, but the website was laggy. Thank you.
No issues, it's fine. Like you should be able to do it later on, and thanks a lot. Like because I think you were one of the only people who was unmuting actually, and like you gave some pretty good ideas also for our project. So that's like thanks a lot. So yeah.
Oh, and another concept related to biomineralization is biosedimenting. You might want to look into that, microbial mats and all of that.
Okay, I'll have a look at that. I think I've heard of it, but yeah, I'll have a look at that properly once.
Okay, I'll take my leave now. Thank you once again.
Yes, thanks a lot. Okay. I'll also end the meeting now. So if anybody has any doubts, just feel free to reach out. And we will be sharing the resource material and assignments shortly. Okay. So there's two assignments. Just a reminder, two assignments. Both are on the same link. Assignment one is for day two, and assignment two is based on the coding session today. Okay. So thanks a lot. I'm ending it now. Bye, everyone.